Hello and welcome to our short presentation about pulsed coupling. Today we're going to talk about phase response curves, phase transition curves and Poincaré maps. Until now we have mostly looked at dynamical systems in isolation. In nature, however, most often they do not occur in isolation but in combination and interaction with other dynamical systems. Those interacting systems are referred to as coupled systems. One example of coupled systems that we all know are neurons that communicate with other neurons. In order to understand how coupled systems work in detail, it is essential to understand first how a single dynamical system reacts to an external input such as pulses. This is why we focus on pulsed coupling in this video. More specifically, our goal is to describe the effect that a pulse has an oscillatory dynamical system as simple as possible. We will do that by describing the effect of the pulse as the relative change in the phase of the system. But what is a phase? A phase refers to the fraction of the oscillation of the system that is covered up to time t. So one full oscillation or turn around the limit cycle corresponds to one full phase capital T. All other points in time are described as a fraction of capital T. As every point on the limit cycle corresponds to a phase, we have a mapping between the phase and the position on the limit cycle. Because of that, we can describe the position of our system in terms of a phase, rather than in terms of time. Now, how can we describe the effect of the pulse in terms of phase? This is where we come to the topic of phase response curves. As you can see here, the system is pushed away from the limit cycle by the pulse, so it is not mapped to a phase. We know, however, that it will go back to the limit cycle eventually, as the limit cycle attracts all trajectories in its proximity, as indicated by the light blue trajectory. So we can mark the point at which the system is back on the limit cycle as our new phase, the phase after the impact of the pulse. We can also mark the point at which the system would be without the pulse after the same amount of time. This is the old phase, the yellow trajectory. And finally, we can describe the effect of the pulse as the shift in a phase that it caused, marked here in green. This shift in phase is already our phase response curves. In this graphic, the shift is represented by the green area. We can compute the PCR by subtracting the old phase Q from the new phase Q new. When we do that for every phase Q, we can represent the response behavior of the system as a curve. The curve shows how the system reacts to the pulse, independent of the current phase. In this example, a pulse has the biggest effect when it occurs roughly at the middle of the phase. All of this is interesting for us because it can be used as a measure of the effect a given stimulus has on the timing of action potentials. PRCs therefore model neurons' periodic firing behavior based on a given stimulus. So this is all interesting, but let's take a look at how science used this method. All papers we're talking about can be found at the end of our presentation. What you can see here is a cross-section of the human brain. The area of interest is the globus pallidus. Researchers use PRC to look at oscillations between neural networks inside of it. They could observe how certain inputs lead to delayed spiking and also gain other insight on the globus pallidus. This is of major interest to us regarding we are talking about the area supposedly responsible for controlling consciousness. But there's more. This is a Purkinje cell. It sits in the cortex of our cerebellum and is responsible for the computation and information processing. Italian researchers used PRC to analyze it and found how the cell switches between being phase-dependent and phase-independent. So you see, there's actual usage in modern neuroscience for this technique. So brace yourself, because we're going to show you 2 more techniques of that kind.
So, as we explained priorly, the phase response curve explains the phase change due to a stimulus. So, how a phase responds to a pulse? The phase transition curve, in contrast, shows us the effect or the result after the perturbation. So the phase transition curve is just a cumulative sum of the phase response curve. To see the effect, we need to compare our curve to the diagonal. If the curve is equal to the diagonal, it means that there is no effect. If our curve is over the diagonal, our phase is being sped up. If the curve is under the diagonal, that means our phase is being slowed down. So, as you can see, PTCs are used for similar models compared to phase response curves. The main difference is when to use which. The phase transition curves are more convenient when the phase shifts are small so they can be magnified. And the phase transition curves, in contrast, are better when phase shifts are large and comparable with the period of oscillation. As with phase response curves, this is still used in neuroscience. For example, researchers of the Institute for Biological Studies in California used PTC to prove that the amount of neurons influences the interconnectivity between them. They concluded this interconnectivity to be the primary reason for neural maps. Another interesting paper we have linked for you shows how the amount of transitions optimizes the processing of cells. Even though this is no shockingly new finding, it's always interesting how different techniques lead to the same solution.
So, let's talk about Poincaré return maps. To understand Poincaré return maps, you have to know about Floquet theory. Floquet theory is a branch of the theory of ordinary differential equations. It's the class of solutions to periodic linear differential equations. Furthermore, it explains the stability type of periodic systems. These periodic systems are of the form x t plus t equals a of t times x. This extends to the form p of t times the exponential. In this form, p of t describes the periodic component of the orbit and the exponential term describes the stability of said orbit. This leads us to the different periodic solutions of flow maps. We have two stability types for periodic orbits: the stable periodic orbit and the unstable periodic orbit. In a stable periodic orbit, the next iteration p of n plus 1 is equal to p of n. This just describes the trajectory coming back to the same point in the map, therefore the orbit converges. In an unstable periodic orbit, p of n plus 1 is not equal to p of n. It either gets smaller on every iteration or larger. Its trajectory never hits the same point on the map again, therefore we can say the orbit diverges. Let's then look at Poincaré maps. Poincaré maps are used to describe the behavior of complex nonlinear high-dimensional systems. By defining a lower-dimensional subspace, you can track the intersections of the periodic orbit. For example, you would use a two-dimensional subspace for a three-dimensional system. This subspace is called the Poincaré section. By tracking these intersections, we can generate a discrete collection of intersections which can be used for further analysis. Basically, Poincaré maps generalize the Floquet theory by also using the next time step x of n plus 1 and looking at its behavior. If x of n plus 1 ends up at the same point again, the solution converges and we have a stable periodic orbit. If it shifts, we have an unstable orbit. This can be very useful as sometimes higher dimensional complex systems can be hard to analyze, and by defining a lower dimensional subspace, it gets easier and more understandable. To visualize Poincaré maps, I'm going to show you how they can be applied in a well-known neuron model. I'm sure all of you remember the Hodgkin-Huxley model. It explains mechanisms underlying the firing behavior of a neuron, which requires non-local bifurcation analysis. Examining Poincaré maps is a state-of-the-art technique to achieve this. The last paper we're going to present to you is about researchers who have used Poincaré maps to look at cell development. This new technique was also used to inspect neurons. Beforehand, techniques relied on Euclidean geometry. However, Poincaré maps allow us to transform complex data into two-dimensional space without using Euclidean geometry while still keeping the original distances. So you see all the techniques displayed today show real-life applications which you might stumble upon again during your ongoing neuroscience career.